As you settle into this moment, wherever you find yourself, notice the rhythm that has been with you since your very first instant of life. The gentle rise and fall of your chest, the quiet passage of air through your body. Right now, without any conscious effort, your respiratory system is performing one of nature's most essential and elegant processes. You are breathing. In this present moment, oxygen molecules are crossing into your bloodstream. Carbon dioxide is being released, and an intricate network of passages, surfaces, and vessels is sustaining your existence with remarkable precision. Tonight, we explore the respiratory system, a beautiful architecture of airways and exchange surfaces that connects you to the atmosphere surrounding our planet. This is not merely a collection of organs, but rather a sophisticated interface between your internal world and the external environment. Every breath you take represents millions of years of evolutionary refinement, a system so finely tuned that it adjusts moment by moment to your body's changing needs without requiring any conscious direction from you. We will move through this subject gently, discovering how air becomes life, how simple gases enable complex thought, and how your body maintains the delicate balance of respiration even as you rest. This is a journey through structures both large and microscopic, through processes both mechanical and chemical, through systems that work in concert to sustain every cell in your body. We are not here to memorize lists or prepare for examination, but rather to appreciate the quiet intelligence of biological design. Think of the respiratory system as a great river delta where countless streams and channels converge and diverge, where boundaries between air and blood become thin as gossamer, where exchange happens across surfaces vast enough to cover a tennis court yet folded compactly within your chest. Throughout this exploration, we will return again and again to themes of surface area, of gradients, of the wisdom inherent in structures that have evolved to maximize efficiency while maintaining resilience. You may notice, even now, the automatic nature of your breathing. Perhaps your breath has deepened slightly as your attention turns toward it. This awareness itself demonstrates something profound about the respiratory system. It operates continuously in the background of consciousness, yet can be brought into voluntary control at any moment. It bridges the autonomic and the voluntary, the unconscious and the conscious, serving as a tangible connection between mind and body. Let us begin gently with a question that seems simple but opens into complexity. What exactly is respiration? The answer reveals itself in layers, each more detailed than the last, each showing us a different aspect of this fundamental process. At its most basic level, respiration refers to the exchange of gases between an organism and its environment. For humans and most animals, this means taking in oxygen from the air and releasing carbon dioxide as a waste product of metabolism. But this simple definition barely scratches the surface of what is actually happening. The term respiration actually encompasses two distinct but interconnected processes. External respiration describes the exchange of gases between the air and the blood. This is the breathing we are most familiar with, the movement of air into and out of the lungs, the transfer of oxygen into blood vessels and carbon dioxide out of them. It involves mechanical work, the expansion and contraction of the chest, the flow of air through branching passages, the crossing of gases across delicate membranes. Internal respiration, sometimes called cellular respiration, describes the exchange of gases between the blood and the tissues of the body. and the subsequent use of oxygen within cells to generate energy. This is the ultimate purpose of breathing, to deliver oxygen to mitochondria within cells, where it participates in the remarkable process of converting nutrients into usable energy in the form of ATP, carbon dioxide, produced as a byproduct of this energy production, then travels the reverse journey from cells to blood to lungs to atmosphere. These two forms of respiration are inseparably linked. External respiration serves internal respiration. The breath you take right now begins a cascade that ends with ATP being produced in a neuron in your brain, in a muscle cell in your heart, in every living cell throughout your body. The respiratory system is therefore not just about breathing, it is about energy, about life itself at the cellular level. The importance of this system becomes apparent when we consider what happens in its absence. Brain cells begin to die within minutes without oxygen. The heart cannot maintain its rhythm. Consciousness fades. Yet despite this critical importance, the respiratory system performs its duties so reliably, so quietly, that we can go hours, days, even years without giving it conscious thought. It is always there, this faithful rhythm, this constant exchange. Understanding the respiratory system also means understanding its relationship to other body systems. It works in intimate partnership with the cardiovascular system, which transports the gases the lungs exchange. It responds to signals from the nervous system, which monitors carbon dioxide levels and adjusts breathing rate accordingly. It influences and is influenced by the acid-base balance of the blood, playing a crucial role in maintaining the narrow pH range that cellular chemistry requires. There is something almost musical about respiration when viewed as a whole. Different structures play different notes. Different processes occur at different tempos, yet all combine into a harmonious whole that sustains life. The rhythmic expansion of the lungs, the steady diffusion of gases, the constant adjustments of breathing rate and depth. These create a symphony that has played without interruption since the moment of your birth. Now, let us move deeper into the territory of the respiratory system by examining its fundamental building blocks. Every complex system can be understood by first grasping its components, and the respiratory system is composed of distinct anatomical structures, each designed for specific functions, all working together seamlessly. The respiratory system can be divided conceptually into two main zones, the conducting zone and the respiratory zone. This division reflects function. The conducting zone consists of passages that transport air but do not participate in gas exchange. The respiratory zone consists of structures where actual gas exchange occurs. Understanding this distinction helps clarify why different parts of the respiratory tract have different structural features. The conducting zone begins with the nose and mouth, the external openings through which air enters the body. These are not merely holes but sophisticated portals. The nasal cavity, with its convoluted turbinate bones covered in mucous membrane, warms and humidifies incoming air. It also filters particulate matter, trapping dust and microorganisms in sticky mucus before they can reach deeper structures. The nose is quite literally an air conditioning system, bringing atmospheric air closer to body temperature and humidity before it reaches the delicate lung tissue. From the nasal cavity or mouth, air travels to the pharynx, a shared passage for both air and food. This region represents an evolutionary compromise. The pharynx serves dual purposes, which is efficient but also creates the possibility of choking. The larynx, positioned at the top of the trachea, serves as a guardian here. During swallowing, a flap of cartilage called the epiglottis folds down over the laryngeal opening, directing food to the esophagus behind while protecting the airway in front. The larynx itself is more than a protective valve. It is the organ of voice, containing the vocal cords or vocal folds. These are bands of tissue that vibrate as air passes through them, producing sound waves that are then shaped by the tongue, lips, and palate into speech. The respiratory system thus gives us not only life but also the capacity for complex communication, for song, for the expression of thought through spoken language. Below the larynx lies the trachea, commonly called the windpipe. This is a tubular structure about 10 to 12 centimeters long, reinforced with C-shaped rings of cartilage. These cartilage rings are crucial. They prevent the trachea from collapsing when air pressure inside drops during inhalation, maintaining an open airway at all times. The open part of the C faces backward toward the esophagus, allowing that structure to expand during swallowing without being restricted by rigid cartilage. The inner surface of the trachea is lined with a specialized epithelium, a tissue type that appears throughout the conducting zone. This epithelium contains goblet cells, which secrete mucus, and ciliated cells, which bear tiny hair-like projections called cilia. The cilia beat in coordinated waves, always moving upward, creating what is sometimes called the mucociliary escalator. Mucus traps particles and pathogens, and the cilia sweep this mucus upward toward the pharynx, where it can be swallowed or expectorated. This is one of the respiratory system's primary defense mechanisms, a constant cleansing process that protects the delicate lower airways. The trachea divides at a point called the carina into two primary bronchi, one entering each lung. This division is the beginning of the branching architecture that defines the respiratory tree. The bronchi continue to divide, becoming smaller and more numerous with each generation of branching. Primary bronchi become secondary bronchi, also called lobar bronchi because each supplies a lobe of the lung. Secondary bronchi divide into tertiary bronchi, also called segmental bronchi, which supply specific bronchopulmonary segments. This pattern of division continues through many generations. Each division point is called a bifurcation, and the passages become progressively smaller in diameter while increasing dramatically in total number. By the time we reach the smallest conducting airways, called terminal bronchioles, we have gone through approximately 16 to 18 generations of branching, and there are tens of thousands of these tiny passages. This repeated branching creates an enormous total cross-sectional area, much like how a river delta spreads water across many channels. As the airways branch and become smaller, their structure changes in predictable ways. The larger bronchi, like the trachea, are supported by cartilage, though the cartilage appears as irregular plates rather than complete rings. The walls contain smooth muscle, which allows these passages to dilate or constrict in response to various signals. The epithelium remains ciliated and mucus secreting, continuing the cleansing function. In smaller bronchi and bronchioles, cartilage disappears entirely. These airways are held open primarily by the elastic recoil of surrounding lung tissue and by the natural tendency of the airways to be pulled outward as the lungs expand. Smooth muscle becomes proportionally more prominent in the walls of bronchioles, giving these structures significant ability to change diameter. The epithelium becomes simpler with fewer ciliated cells and goblet cells, eventually transitioning to a simple cuboidal epithelium in the smallest airways. This structural progression reflects functional changes. The larger airways primarily conduct and condition air. The smaller airways, with their abundant, smooth muscle and lack of cartilage, are the primary site of airway resistance and also the structures most affected by conditions like asthma, where smooth muscle constriction can narrow the passages significantly. The terminal bronchioles represent the end of the conducting zone. Beyond them lies the respiratory zone, where the character of the airways changes fundamentally. Terminal bronchioles give rise to respiratory bronchioles, which are transitional structures. They still conduct air, but their walls begin to feature tiny outpouchings called alveoli, where gas exchange can occur. Respiratory bronchioles lead to alveolar ducts, which are essentially tubular passages whose walls consist entirely of alveoli. Finally, alveolar ducts end in alveolar sacs, clusters of alveoli resembling bunches of grapes. The alveoli themselves are the fundamental functional units of gas exchange. Each alveolus is a small, thin-walled chamber, roughly hemispherical in shape. The human lungs contain approximately 300 million alveoli, creating a gas exchange surface area of about 70 to 80 square meters, roughly the size of a tennis court. This immense surface area, folded and packed into the volume of your chest cavity, is one of the respiratory system's most remarkable features. The wall of an alveolus is called the alveolar capillary membrane or respiratory membrane. This is where oxygen and carbon dioxide actually cross between air and blood. The membrane is extraordinarily thin, averaging less than 1 micrometer in thickness in many places, bringing air and blood into the closest possible proximity while maintaining a barrier between them. This thinness is essential for efficient diffusion, which we will explore in more detail shortly. The alveolar wall consists primarily of 2 cell types. Type Y alveolar cells, also called type I pneumocytes, are extremely flat cells that cover about 95% of the alveolar surface area. These cells are the ones responsible for gas exchange, their thinness allowing rapid diffusion. Type 2 alveolar cells, or type 2 pneumocytes, are smaller, more cuboidal cells that occupy only about 5% of surface area, but are numerically more common. Type 2 cells serve a crucial function. They produce and secrete pulmonary surfactant. Surfactant is a complex mixture of lipids and proteins that spreads across the inner surface of alveoli, reducing surface tension. Without surfactant, the tendency of water molecules at an air-water interface to cohere would cause alveoli to collapse, particularly during exhalation when lung volume decreases. Surfactant prevents this collapse, allowing alveoli to remain open and functional. The importance of surfactant becomes tragically clear in premature infants whose type 2 cells have not yet matured enough to produce adequate amounts, leading to respiratory distress syndrome. Between adjacent alveoli are thin partitions called alveolar septa, and within these septa runs an incredibly dense network of capillaries. These are the smallest blood vessels, only 1 cell thick, arising from branches of the pulmonary arteries and draining into tributaries of the pulmonary veins. The capillaries are so numerous and so closely packed that blood flows through the lungs in what amounts to an almost continuous sheet, maximizing contact with air-filled alveoli. This architecture, millions of thin-walled air sacs surrounded by a dense capillary network, represents the culmination of the respiratory system's design. It maximizes surface area, minimizes diffusion distance, and creates optimal conditions for gas exchange. The conducting airways have brought air to this point, warmed it, humidified it, cleaned it, and distributed it throughout the lung. Now, in the respiratory zone, the actual purpose of breathing is fulfilled. Oxygen enters the blood, carbon dioxide leaves it. Now, let us consider how these structures are organized into the complete organs we call the lungs. The lungs are paired organs occupying the thoracic cavity, the space within the chest bounded by the ribs, sternum, and vertebral column above and by the diaphragm below. They are separated from each other by the mediastinum, a central compartment containing the heart, great vessels, esophagus, and other structures. Each lung has a somewhat conical shape, with a rounded apex extending slightly above the first rib and a concave base resting on the diaphragm. The right and left lungs are not identical. The right lung is larger, accounting for about 55% of total lung volume, and is divided into 3 lobes: superior, middle, and inferior, separated by deep fissures. The left lung is smaller, making room for the heart, which extends more to the left side of the chest and contains only 2 lobes, superior and inferior. The left lung also features a pronounced indentation on its medial surface called the cardiac notch, an accommodation for the heart. Each lung is enclosed in a double-layered membrane called the pleura. The visceral pleura adheres tightly to the lung surface, following every contour and extending into the fissures between lobes. The parietal pleura lines the inner surface of the chest wall, the diaphragm, and the mediastinum. Between these 2 pleural layers is the pleural cavity, a potential space containing a thin film of pleural fluid. The pleural arrangement is functionally crucial. The pleural fluid acts as a lubricant, allowing the lungs to slide smoothly against the chest wall during breathing movements. More importantly, the fluid creates surface tension that holds the visceral and parietal pleura together, much like 2 glass slides with water between them stick together while still being able to slide across each other. This coupling means that when the chest wall expands, the lungs are pulled along with it, and when the chest wall recoils, the lungs do the same. The pleural cavity normally contains only about 15 milliliters of fluid and maintains a pressure slightly below atmospheric pressure, typically around -4 to -8 centimeters of water at rest. This negative intrapleural pressure is also important for keeping the lungs expanded. The lungs have natural elastic recoil due to elastic fibers in their tissue and surface tension at alveolar surfaces. This elastic recoil creates an inward pull, a tendency to collapse. The negative intrapleural pressure provides an outward pull that counterbalances this, keeping the lungs inflated even at the end of a normal exhalation. If air enters the pleural cavity, a condition called pneumothorax, this carefully maintained negative pressure is lost. The lung on the affected side can then collapse as its elastic recoil goes unopposed. This demonstrates how important the sealed pleural space is to normal respiratory mechanics. The integrity of this space is maintained by the continuous absorption of fluid and any gases that might enter, keeping the pleural cavity functionally closed. The lungs are highly vascular organs, receiving blood through 2 separate circulations. The pulmonary circulation brings deoxygenated blood from the right ventricle of the heart via the pulmonary arteries. This blood flows through the pulmonary capillaries surrounding the alveoli, where it releases carbon dioxide and picks up oxygen, then returns to the left atrium via the pulmonary veins. This is the circulation directly involved in gas exchange. The bronchial circulation arising from the aorta supplies oxygenated blood to the lung tissue itself, to the bronchi, bronchioles, visceral pleura, and other supporting structures. This blood nourishes the lung tissue in the same way that systemic arteries nourish other organs. Most bronchial venous blood drains into pulmonary veins rather than returning to the right heart, creating a small amount of mixing between oxygenated and deoxygenated blood. The lungs are also extensively innervated by both divisions of the autonomic nervous system. Parasympathetic fibers from the vagus nerve cause bronchoconstriction and increase mucus secretion. Sympathetic fibers cause bronchodilation and decrease secretions. This autonomic control allows breathing to be adjusted according to metabolic needs and in response to various stimuli. Sensory nerves provide information about lung stretch, chemical irritants, and other factors that influence breathing patterns. Understanding this hierarchical organization from the whole lungs down through lobes, bronchopulmonary segments, lobules, and ultimately to individual alveoli gives us a framework for understanding respiratory function and pathology. The lungs are not homogeneous masses, but rather precisely organized structures where each level of hierarchy serves specific purposes. Having established the anatomy, we can now explore the mechanics of breathing itself, the physical processes that move air into and out of the lungs. Breathing, or pulmonary ventilation, requires pressure changes within the thoracic cavity. Air flows from regions of higher pressure to regions of lower pressure, a principle that governs all the movement of air through the respiratory system. At rest, between breaths, the pressure inside the lungs equals atmospheric pressure. For air to flow into the lungs, the pressure inside must become less than atmospheric. For air to flow out, pressure inside must exceed atmospheric pressure. These pressure changes are created not by the lungs themselves, which are passive structures, but by the muscles that change the volume of the thoracic cavity. Inhalation, or inspiration, is an active process requiring muscular contraction. The primary muscle of inspiration is the diaphragm, a dome-shaped muscle that forms the floor of the thoracic cavity. When the diaphragm contracts, it flattens and descends, increasing the vertical dimension of the thoracic cavity. This single muscle is responsible for about 75% of the volume change during quiet breathing. Simultaneously, the external intercostal muscles, which run between the ribs in a downward and forward direction, contract. This pulls the ribs upward and outward in a motion sometimes compared to lifting a bucket handle. This increases both the anteroposterior and lateral dimensions of the thoracic cavity. Together, the diaphragm and external intercostals increase thoracic volume in all three dimensions. As thoracic volume increases, the parietal pleura moves with the chest wall and diaphragm. The visceral pleura, coupled to the parietal pleura by pleural fluid, pulls the lung surface outward. The lungs expand, increasing the volume of the alveolar spaces. According to Boyle's law, if volume increases while the amount of gas remains constant, pressure decreases. The pressure inside the alveoli drops to about 1 to 3 millimeters of mercury below atmospheric pressure. This small pressure difference is sufficient to cause air to flow into the lungs from the atmosphere. Air enters through the nose or mouth and flows down the pressure gradient through the conducting airways until it reaches the alveoli. Inhalation continues until pressures equalize, at which point airflow stops. The lungs are now expanded, holding a larger volume of air than they did before the breath began. Exhalation or expiration during quiet breathing is primarily a passive process. The muscles that drove inspiration simply relax. The diaphragm returns to its dome shape, the ribs move downward and inward, thoracic volume decreases. The lungs, which were stretched during inspiration, recoil elastically, like a stretched rubber band returning to its original length. This elastic recoil is due both to elastic fibers in lung tissue and to surface tension in alveoli. As lung volume decreases, alveolar pressure rises to about 1 to 3 millimeters of mercury above atmospheric pressure, creating a pressure gradient in the opposite direction. Air flows out of the lungs, through the conducting airways, and into the atmosphere. Exhalation continues until pressures once again equalize, with the lungs now at their resting volume. This cycle repeats 12 to 20 times per minute in a resting adult, each breath bringing in and expelling roughly 500 milliliters of air, called the tidal volume. The process is so smooth, so automatic, that it continues whether you are aware of it or not, adjusting moment by moment to keep pace with your body's changing oxygen demands and carbon dioxide production. During exercise or other circumstances requiring greater ventilation, breathing becomes deeper and more forceful. Inspiration becomes more active, recruiting accessory muscles that can further expand the thoracic cavity. The sternocleidomastoid muscles can lift the sternum, the scalene muscles can elevate the first two ribs, and the pectoralis minor can raise ribs 3 through 5. These muscles, which are not normally active during quiet breathing, can substantially increase thoracic expansion. Expiration too can become active when needed. The internal intercostal muscles, oriented differently from the external intercostals, pull the ribs downward and inward more forcefully than passive recoil alone. The abdominal muscles, rectus abdominis, external and internal obliques, and transversus abdominis, can contract to push the abdominal contents upward against the diaphragm, forcing it higher into the thoracic cavity and dramatically reducing lung volume. These active mechanisms allow forced exhalation to expel much more air than passive expiration. The volumes and capacities of the lungs can be measured and quantified, providing useful information about respiratory function. Tidal volume, as mentioned, is the volume inhaled or exhaled during normal quiet breathing, typically about 500 milliliters. But the lungs can hold much more air than this. After a normal tidal inspiration, it is possible to inhale even more deeply. This additional volume that can be inspired beyond tidal volume is called the inspiratory reserve volume, typically about 3,000 milliliters in males, somewhat less in females. Similarly, after a normal tidal expiration, it is possible to exhale more forcefully. This additional volume that can be expired beyond tidal volume is the expiratory reserve volume, about 1,100 milliliters. Even after the most forceful expiration, some air remains in the lungs. This residual volume, about 1,200 milliliters, is air that cannot be voluntarily expelled. It remains in the airways and alveoli, keeping these structures partially inflated even at maximal exhalation. The presence of residual volume means that even at the end of expiration, the lungs are not empty, but rather at a reduced volume. Various combinations of these volumes are given specific names. Inspiratory capacity is tidal volume plus inspiratory reserve volume, the maximum amount that can be inhaled starting from resting expiratory level. Functional residual capacity is expiratory reserve volume plus residual volume, the amount of air remaining in the lungs at the end of a normal quiet expiration. Vital capacity is inspiratory reserve volume plus tidal volume plus expiratory reserve volume, the maximum volume that can be moved into or out of the lungs in a single breath. Total lung capacity is the sum of all volumes, the total amount of air the lungs can hold. These measurements vary with body size, sex, age, and physical fitness, but they provide a baseline for assessing respiratory function. Conditions that restrict lung expansion reduce vital capacity. Conditions that trap air in the lungs increase residual volume. Spirometry, the measurement of these volumes and the rate at which air can be moved, is a fundamental tool in respiratory medicine. Understanding lung mechanics also requires considering the resistance to airflow. As air moves through the conducting airways, it encounters resistance due to friction between air molecules and airway walls. The total resistance of the respiratory system depends primarily on the diameter of the airways, particularly the medium-sized bronchi and bronchioles. Because resistance varies inversely with the fourth power of radius, even small changes in airway diameter can significantly affect resistance. Airway diameter is not fixed, but can change in response to various factors. Smooth muscle in the walls of bronchi and bronchioles can contract or relax, changing airway caliber. Parasympathetic stimulation causes bronchial smooth muscle to contract, narrowing airways and increasing resistance. Sympathetic stimulation and circulating epinephrine cause smooth muscle relaxation, widening airways and decreasing resistance. During exercise, sympathetic activation causes bronchodilation, reducing airway resistance and facilitating increased ventilation. Inflammation can also affect airway diameter. Swelling of airway walls, excess mucus production, and contraction of smooth muscle all contribute to increased resistance in conditions like asthma and chronic obstructive pulmonary disease. The respiratory system normally maintains relatively low resistance, but when resistance increases significantly, breathing requires more effort and may become noticeably difficult. Lung compliance is another important mechanical property referring to how easily the lungs expand in response to pressure changes. Compliance is a measure of distensibility, how much volume change occurs for a given pressure change. High compliance means the lungs expand easily. Low compliance means they are stiff and resistant to expansion. Normal lungs have relatively high compliance, expanding readily as thoracic volume increases. Several factors can reduce compliance. Fibrosis, where lung tissue is replaced by scar tissue, makes lungs stiffer. Deficiency of surfactant increases surface tension, making alveoli harder to inflate. Pulmonary congestion or edema increases tissue resistance to expansion. Reduced compliance means that more muscular effort is required to achieve the same volume of air movement, making breathing laborious. The work of breathing is normally quite low, consuming only about 3 to 5% of total oxygen consumption at rest. This work includes overcoming elastic resistance of the lungs and chest wall, overcoming airway resistance, and overcoming inertia of the respiratory system. During quiet breathing, the work is minimal and accomplished unconsciously. During exercise or in disease states, the work of breathing can increase substantially, sometimes to the point where it consumes a significant fraction of available oxygen. As we move through this exploration of the respiratory system, we are building an understanding of how structure serves function. The branching airways distribute air. The thin alveolar capillary membrane facilitates diffusion. The pleural space couples lung movement to chest wall movement. The respiratory muscles create the pressure changes that drive ventilation. Each element has its purpose, each contributes to the whole. The respiratory system's mechanical aspects are governed by physical principles, pressure gradients, gas laws, fluid dynamics. Yet these physical processes are not random but highly organized, coordinated by control systems we will explore shortly. The body knows when to breathe faster or slower, deeper or more shallowly. It adjusts breathing to maintain precise chemical balance in the blood. This regulation operates largely outside conscious awareness, another example of the respiratory system's role as a bridge between voluntary and involuntary function. As we prepare to delve deeper into gas exchange and regulation, notice once more your own breathing, the air moving in and out, the subtle expansion and relaxation of your chest. This is not merely a mechanical process, but a moment-by-moment conversation between your body and the atmosphere, between your cell's needs and the respiratory system's elegant responses. This rhythm has continued since you began reading, adjusted without your conscious input, and will continue as we move forward, exploring how this system maintains the delicate balance of gases that life requires. We continue now from those foundations, from the structures and mechanics we have explored, moving into the chemistry and physics that transform breathing into life. The air has entered your lungs, expanded the alveoli, brought atmospheric gases into contact with the blood flowing through pulmonary capillaries. Now we examine what happens at that thin membrane where air meets blood, where the actual exchange occurs that justifies all the anatomical complexity we have discussed. Gas exchange is fundamentally a process of diffusion, the passive movement of molecules from areas of higher concentration to areas of lower concentration. This seemingly simple principle drives one of biology's most critical functions. Diffusion does not require energy, does not require pumps or active transport mechanisms. It happens spontaneously, driven by the random motion of molecules and the statistical tendency for concentrations to equalize across a permeable barrier. The respiratory membrane across which this diffusion occurs consists of several layers, yet remains remarkably thin. Starting from the alveolar side, we find a layer of surfactant and fluid lining the alveolus. Then comes the alveolar epithelium, consisting primarily of those extremely flat type I pneumocytes. Beneath this lies a basement membrane, sometimes fused with the capillary basement membrane. Then the capillary endothelium, the single-cell thick wall of the blood vessel. Finally, the blood plasma and the membrane of the red blood cell itself. All these layers together average less than 1 micrometer in thickness, and in many places are even thinner. This minimal diffusion distance is crucial because the rate of diffusion is inversely proportional to distance. Making the membrane 10 times thinner increases diffusion rate tenfold. The respiratory membrane represents an extraordinary compromise between thinness for rapid diffusion and structural integrity for maintaining the separation between air and blood. The driving force for diffusion is the partial pressure gradient. In a mixture of gases, each gas exerts a partial pressure proportional to its percentage of the total. In atmospheric air at sea level, where total pressure is 760 millimeters of mercury, oxygen comprises about 21% of the mixture and therefore exerts a partial pressure of about 160 millimeters of mercury. Nitrogen, making up about 78%, exerts about 600 millimeters. Carbon dioxide, present in only 0.04%, exerts a mere 0.3 millimeters. When air is inhaled, it becomes humidified in the upper respiratory tract, and water vapor now contributes to total pressure, diluting the other gases slightly. By the time inspired air reaches the alveoli, it has also mixed with the residual volume remaining from previous breaths. The alveolar air therefore has lower oxygen partial pressure than atmospheric air, typically about 100 to 105 millimeters of mercury, and higher carbon dioxide partial pressure, about 40 millimeters of mercury, due to the carbon dioxide continually entering alveoli from the blood. Blood arriving at the lungs in the pulmonary capillaries has just returned from the systemic tissues, where it released oxygen and picked up carbon dioxide. Venous blood entering the pulmonary capillaries has an oxygen partial pressure of about 40 millimeters of mercury and a carbon dioxide partial pressure of about 46 millimeters of mercury. These values create the gradients that drive diffusion. For oxygen, alveolar partial pressure of 100 millimeters exceeds venous blood partial pressure of 40 millimeters, creating a gradient of 60 millimeters. Oxygen diffuses from alveolar air into the blood across the respiratory membrane. This diffusion is rapid and efficient. Under normal resting conditions, blood spends about 3/4 of a second traversing a pulmonary capillary. Oxygen equilibration between alveolar air and blood occurs in about 1/4 of a second, leaving substantial reserve time. For carbon dioxide, the gradient is smaller but in the opposite direction. Venous blood partial pressure of 46 millimeters exceeds alveolar partial pressure of 40 millimeters, a gradient of only 6 millimeters. Despite this smaller gradient, carbon dioxide diffuses readily from blood into alveolar air because carbon dioxide is much more soluble in water than oxygen is. This higher solubility compensates for the smaller pressure gradient, and carbon dioxide equilibration also occurs within the time blood spends in the pulmonary capillaries. By the time blood exits the pulmonary capillaries, it has been transformed. Oxygen partial pressure has risen from 40 to 100 millimeters of mercury. Carbon dioxide partial pressure has fallen from 46 to 40 millimeters. This oxygenated blood returns to the left atrium, then to the left ventricle, then is pumped out through the aorta to the systemic circulation, carrying its oxygen to tissues throughout the body. At the tissue level, the process reverses. Metabolically active cells consume oxygen and produce carbon dioxide. The partial pressure of oxygen in tissue cells is low, perhaps 40 millimeters or less. The partial pressure of carbon dioxide is high, perhaps 46 millimeters or more. When arterial blood with oxygen partial pressure near 100 millimeters reaches tissue capillaries, oxygen diffuses down its gradient from blood into the tissues. When blood with carbon dioxide partial pressure of 40 millimeters encounters tissues where carbon dioxide partial pressure exceeds 46 millimeters, carbon dioxide diffuses from tissues into blood. This internal respiration at the tissue level is the mirror image of external respiration at the lungs. The two processes are linked by the circulation, with the heart providing the pumping force that circulates blood between lungs and tissues. The respiratory and cardiovascular systems form an integrated unit for gas transport. Neither system alone suffices. Together, they create a continuous loop of oxygen delivery and carbon dioxide removal. The efficiency of gas exchange depends on several factors working in concert. Adequate ventilation brings fresh air to the alveoli, maintaining appropriate partial pressures. Adequate perfusion brings blood to the pulmonary capillaries at a rate matching ventilation. Adequate diffusion capacity determined by membrane thickness, surface area, and gas solubility, allows rapid equilibration. These three factors, ventilation, perfusion, and diffusion, must be balanced for optimal gas exchange. Ventilation-perfusion matching is a particularly important concept. Ideally, areas of the lung receiving more blood flow should also receive more ventilation, and vice versa. Perfect matching would mean every alveolus receives ventilation and perfusion in ideal proportion. In reality, some mismatch occurs even in healthy lungs due to gravity's effects on blood flow and regional differences in lung expansion. Blood flow in the lungs is gravity dependent. In an upright person, perfusion is greatest at the lung bases and lowest at the apices. This occurs because the pulmonary circulation is a low-pressure system, and the weight of the blood column creates pressure differences between the top and bottom of the lungs. Ventilation also varies with position, but to a lesser degree, with bases receiving somewhat more ventilation than apices. The ratio of ventilation to perfusion, called the VQ ratio, varies regionally. At the apices, ventilation may exceed perfusion, creating a relatively high VQ ratio. At the bases, perfusion may exceed ventilation, creating a relatively low VQ ratio. The body has mechanisms to minimize these mismatches. Hypoxic vasoconstriction, where pulmonary blood vessels constrict in poorly ventilated regions, redirects blood flow away from underventilated areas. Local control of bronchiolar smooth muscle can redirect airflow as well. Despite these compensatory mechanisms, some VQ mismatch persists, which is why arterial oxygen partial pressure leaving the lungs is slightly less than alveolar oxygen partial pressure. The blood exiting the lungs is a mixture from many different alveoli with varying VQ ratios. This physiologic shunt represents normally functioning lung tissue with slight imperfection in matching. In disease states, VQ mismatch can become severe, significantly impairing oxygenation. Now let us examine how gases are actually transported in the blood, because oxygen and carbon dioxide do not simply dissolve in plasma and circulate freely. Most of each gas is carried in specific forms that enhance transport capacity far beyond what simple dissolution would allow. Oxygen transport centers on hemoglobin, the remarkable protein found in red blood cells. Each hemoglobin molecule consists of 4 polypeptide chains, each chain containing a heme group with an iron atom at its center. Each iron atom can bind 1 oxygen molecule. Since each hemoglobin has 4 heme groups, each hemoglobin molecule can carry 4 oxygen molecules. Red blood cells are packed with hemoglobin, about 280 million molecules per cell. The blood contains about 5 million red blood cells per microliter. This concentration of hemoglobin creates enormous oxygen carrying capacity. While dissolved oxygen in plasma accounts for only about 3% of total oxygen transport, oxygen bound to hemoglobin accounts for about 97%. The binding of oxygen to hemoglobin exhibits a property called cooperativity. When the first oxygen molecule binds to a hemoglobin, it changes the protein's shape slightly, making it easier for the second oxygen to bind. The second oxygen makes the third easier, and the third facilitates the fourth. This cooperative binding creates a characteristic S-shaped oxygen-hemoglobin dissociation curve when hemoglobin saturation is graphed against oxygen partial pressure. At high oxygen partial pressures, such as those in the lungs where alveolar oxygen is around 100 millimeters of mercury, hemoglobin becomes nearly fully saturated, typically about 97 to 98%. Each hemoglobin molecule carries close to its maximum of 4 oxygen molecules. At lower oxygen partial pressures, such as those in resting tissues where oxygen might be 40 millimeters, hemoglobin saturation drops to perhaps 75%. This means each hemoglobin has released, on average, about one of its 4 oxygen molecules. The shape of the dissociation curve is physiologically important. The flat upper portion means that even if alveolar oxygen partial pressure drops somewhat, hemoglobin saturation remains high. You can be at moderate altitude where atmospheric oxygen is reduced or have mild lung disease, and still achieve near normal hemoglobin saturation. The steep middle portion means that in the tissues, small decreases in oxygen partial pressure cause substantial oxygen release from hemoglobin, allowing precise matching of oxygen delivery to tissue needs. Several factors influence the oxygen-hemoglobin relationship, shifting the dissociation curve left or right. Increased temperature, increased carbon dioxide, increased hydrogen ion concentration causing decreased pH, and increased levels of a compound called 2,3-bisphosphoglycerate all shift the curve to the right. A rightward shift means that at any given oxygen partial pressure, hemoglobin holds oxygen less tightly and releases it more readily. This is exactly what happens in metabolically active tissues that are warm, producing carbon dioxide and acids. The conditions that indicate high metabolic activity cause hemoglobin to release oxygen more readily. A beautiful example of physiological adaptation. Conversely, decreased temperature, decreased carbon dioxide, increased pH, and decreased 2,3-bisphosphoglycerate shift the curve leftward, making hemoglobin bind oxygen more tightly. This occurs in the lungs where carbon dioxide is being removed and conditions favor oxygen loading onto hemoglobin. Carbon dioxide transport is more complex than oxygen transport because carbon dioxide exists in blood in 3 distinct forms. About 7% of carbon dioxide is dissolved directly in plasma, a higher percentage than oxygen because of carbon dioxide's greater solubility. About 23% is bound to hemoglobin, though not at the same sites where oxygen binds. Carbon dioxide binds to amino groups on the globin protein chains, forming carbaminohemoglobin. This binding is influenced by oxygen saturation. Deoxygenated hemoglobin binds carbon dioxide more readily than oxygenated hemoglobin, a phenomenon called the Haldane effect. The remaining 70% of carbon dioxide is transported as bicarbonate ion in the plasma. This requires chemical conversion. Carbon dioxide diffusing into blood from tissues enters red blood cells where an enzyme called carbonic anhydrase catalyzes its reaction with water to form carbonic acid. Carbonic acid immediately dissociates into hydrogen ion and bicarbonate ion. The bicarbonate then moves out of the red blood cell into the plasma in exchange for chloride moving in, a process called the chloride shift. In the lungs, these reactions reverse. Bicarbonate enters red blood cells, combines with hydrogen ions to form carbonic acid, which dissociates into carbon dioxide and water. The carbon dioxide diffuses out of the blood into the alveoli and is exhaled. The interconversion between carbon dioxide and bicarbonate is rapid, allowing the blood to carry much more carbon dioxide than simple dissolution would permit. This bicarbonate system is not only a transport mechanism, but also a crucial buffer system. Carbonic acid is a weak acid, and the bicarbonate ion is the conjugate base of this acid. Together, they form one of the body's primary pH buffering systems, helping to maintain blood pH within its narrow normal range of 7.35 to 7.45. The respiratory system plays a central role in pH regulation by controlling carbon dioxide levels, as we shall explore further. The intimate relationship between oxygen and carbon dioxide transport, mediated by hemoglobin and the bicarbonate system, reveals the respiratory system's chemical sophistication. These are not merely mechanical processes of moving air and blood, but carefully orchestrated chemical transformations that maximize gas transport while simultaneously contributing to acid-base balance. The elegance of these mechanisms becomes apparent when we consider how seamlessly they operate, how they automatically adjust to varying conditions, how robust they are across a wide range of physiological states. Now we turn our attention to how breathing is controlled, how the body knows when to breathe faster or slower, deeper or more shallowly. The regulation of respiration is one of the nervous system's most continuous and vital tasks, operating every moment of every day, adjusting ventilation to maintain precise homeostatic balance. The respiratory control centers are located in the brainstem in two main regions. The medullary respiratory center in the medulla oblongata contains groups of neurons that generate the basic rhythm of breathing. Some neurons are active during inspiration, others during expiration. These neurons form interconnected networks that produce rhythmic patterns of activity even in the absence of any sensory input. This intrinsic rhythmicity means that breathing continues automatically without requiring conscious thought. The pontine respiratory centers in the pons help coordinate the transition between inspiration and expiration and can modify the breathing pattern. The pneumotaxic center sends inhibitory signals to the inspiratory neurons in the medulla, limiting inspiration and allowing smooth transition to expiration. The apneustic center sends stimulatory signals that can prolong inspiration. The interplay between these pontine centers and the medullary rhythm generators creates the normal smooth breathing pattern. However, breathing is not rigidly fixed, but continuously adjusted based on sensory feedback. The most important chemical stimuli are carbon dioxide, oxygen, and pH. Of these, carbon dioxide is the primary regulatory signal under normal conditions. Even small increases in blood carbon dioxide levels trigger increased ventilation. This sensitivity to carbon dioxide allows the respiratory system to match ventilation very precisely to metabolic production of carbon dioxide. Central chemoreceptors located in the medulla respond to changes in the pH of cerebrospinal fluid, which in turn reflects changes in blood carbon dioxide levels. Carbon dioxide easily crosses the blood-brain barrier, unlike hydrogen ions, which cross poorly. When blood carbon dioxide rises, carbon dioxide enters the cerebrospinal fluid where it forms carbonic acid, lowering pH. The central chemoreceptors detect this pH drop and stimulate increased ventilation. Peripheral chemoreceptors located in the carotid bodies at the bifurcation of the common carotid arteries and in the aortic bodies in the aortic arch respond to multiple stimuli. They are sensitive to low oxygen partial pressure, high carbon dioxide partial pressure, and low pH. The peripheral chemoreceptors provide the primary stimulus for increased ventilation when oxygen levels fall. They respond within seconds to changes in arterial blood composition, sending signals via cranial nerves to the respiratory centers. Under normal circumstances, arterial oxygen levels remain high enough that oxygen does not drive ventilation. Carbon dioxide is the dominant control. However, if arterial oxygen falls significantly, as might occur at high altitude or in severe lung disease, the peripheral chemoreceptors become powerfully stimulated and drive increased ventilation. This hypoxic drive is particularly important in chronic lung disease, where carbon dioxide levels may be chronically elevated, potentially blunting the normal carbon dioxide response. The respiratory system also responds to pH changes independent of carbon dioxide. Metabolic acidosis, where pH falls due to accumulation of acids other than carbonic acid, stimulates ventilation. The increased breathing blows off carbon dioxide, which shifts the carbonic acid equilibrium and raises pH back toward normal. This respiratory compensation for metabolic acidosis is rapid, beginning within minutes. Similarly, metabolic alkalosis, where pH rises, causes ventilatory depression, allowing carbon dioxide to accumulate and pH to fall back toward normal. Beyond chemical control, mechanoreceptors provide important feedback. Stretch receptors in the smooth muscle of airways respond to lung inflation. When activated by significant lung expansion, they send signals via the vagus nerve to inhibit inspiration, preventing overinflation. This is called the Heering-Breuer reflex. In adults, this reflex is typically active only during deep breathing, but it prevents the lungs from being inflated to volumes that might damage the delicate alveolar structures. Higher brain centers also influence breathing in ways that are not entirely voluntary. Emotional states affect breathing patterns. Anxiety typically increases breathing rate and depth. Calm relaxation slows breathing. The limbic system and hypothalamus can modulate respiratory center activity, creating the breathing changes that accompany different emotional and physiological states. Pain can increase ventilation. Anticipation of exertion can increase breathing even before physical activity begins. Temperature affects breathing as well. Increased body temperature stimulates ventilation, which makes physiological sense because elevated temperature usually indicates increased metabolism and thus increased carbon dioxide production. The hypothalamus integrates temperature information with respiratory control. This multi-layered control system, intrinsic rhythm, chemical feedback, mechanical feedback, voluntary influence, emotional modulation, creates remarkable flexibility and precision. Breathing adjusts moment by moment, breath by breath, maintaining arterial oxygen partial pressure near 100 millimeters of mercury and carbon dioxide partial pressure near 40 millimeters of mercury despite wide variations in metabolic rate and environmental conditions. The normal breathing pattern at rest is regular and relatively effortless, with a rate of 12 to 20 breaths per minute and a tidal volume of about 500 milliliters. This provides a minute ventilation, the total volume breathed per minute of about 6 to 8 liters. During exercise, both rate and depth can increase dramatically, with minute ventilation potentially rising to 150 liters per minute or more in trained athletes. The respiratory system's reserve capacity is substantial. At rest, we use only a small fraction of our maximum ventilatory capacity. This reserve allows for the dramatic increases in ventilation required during exercise. It also provides a buffer against disease. Significant lung damage can occur before resting function is noticeably impaired because the system normally operates well below its maximum capacity. As we explore these regulatory mechanisms, we see the respiratory system not as a passive bellows, but as an actively controlled, continuously adjusted system. The brainstem monitors chemical signals with exquisite sensitivity. The chemoreceptors provide real-time feedback. The system responds appropriately to exercise, emotion, altitude, and metabolic state. This is not simple mechanics, but elegant control theory, implemented in biological hardware. The integration between the respiratory system and other body systems becomes increasingly apparent. The cardiovascular system transports the gases that the respiratory system exchanges. The nervous system controls the respiratory muscles and integrates sensory information. The renal system works alongside the respiratory system in acid-base balance, providing slower but more complete compensation for pH disturbances. The muscular system provides the force for breathing movements. The skeletal system provides the protective and mechanical framework. One can view the body as a city where the respiratory system serves as the interface with the outside world, bringing in one essential resource, oxygen, and expelling one major waste product, carbon dioxide. The cardiovascular system serves as the transportation network, distributing oxygen and collecting carbon dioxide. The cells throughout the body are the individual factories and offices and homes where metabolism occurs, where oxygen is consumed and carbon dioxide is produced. The respiratory control centers are like a utility management system, constantly monitoring demand and adjusting supply. This metaphor extends further. Just as a city has redundant systems and reserve capacity, so does the respiratory system. Just as a city's infrastructure requires maintenance and occasionally needs repair, so do the lungs and airways. Just as a city must respond to changing conditions, rush hour, seasonal variations, special events, the respiratory system must respond to exercise, sleep, altitude, illness. The respiratory system's responsiveness to acid-base balance deserves deeper consideration. pH regulation is critical for life because enzymes and other proteins function properly only within narrow pH ranges. Blood pH is normally maintained between 7.35 and 7.45, an alkaline range. The respiratory system can adjust pH within minutes by changing ventilation, making it the body's first line of defense against pH disturbances. When metabolic processes produce excess acids, as occurs during intense exercise when lactic acid accumulates, blood pH tends to fall. The peripheral chemoreceptors detect this drop and stimulate increased ventilation. The increased breathing eliminates carbon dioxide, which shifts the carbonic acid equilibrium toward the bicarbonate side, consuming hydrogen ions and raising pH back toward normal. This respiratory compensation is rapid and can partially or completely normalize pH within minutes. Similarly, if metabolic processes produce insufficient acid or excess base, pH tends to rise. The respiratory centers respond by decreasing ventilation, allowing carbon dioxide to accumulate. This adds carbonic acid to the blood, lowering pH back toward normal. The respiratory system can thus compensate for both acidosis and alkalosis of metabolic origin. The respiratory system can also cause pH disturbances if ventilation becomes inappropriate. Hyperventilation, breathing more than metabolic needs require, blows off too much carbon dioxide, raising pH and causing respiratory alkalosis. This can occur with anxiety at high altitude where low oxygen stimulates increased breathing, or from voluntary overbreathing. Symptoms may include lightheadedness, tingling in the extremities, and even tetany in severe cases, all due to the effects of elevated pH on nervous system function. Hypoventilation, breathing less than metabolic needs require, allows carbon dioxide to accumulate, lowering pH and causing respiratory acidosis. This can occur with respiratory depression from drugs, neuromuscular disorders affecting breathing muscles, or severe lung disease preventing adequate ventilation. The body attempts to compensate through renal mechanisms, retaining bicarbonate to buffer the excess acid, but this takes days rather than minutes. The intimate relationship between breathing and pH reveals another dimension of respiratory function. It is not only about oxygen delivery, but also about maintaining the chemical environment that all cellular processes require. The respiratory system, cardiovascular system, and renal system together form an integrated pH control network, with the respiratory system providing fast, minute-to-minute adjustments. Throughout all these explorations of gas exchange, transport, and control, notice how the respiratory system exemplifies biological elegance. Simple physical principles, diffusion, pressure gradients, cooperative binding, are harnessed through intricate anatomical structures and sophisticated control mechanisms to achieve reliable performance across a remarkable range of conditions. The system is robust yet flexible, automatic yet controllable, simple in concept yet complex in implementation. The respiratory system operates at multiple scales simultaneously. At the molecular scale, oxygen binds to hemoglobin, carbon dioxide forms bicarbonate. At the cellular scale, type I pneumocytes provide gas exchange surfaces. Type II cells produce surfactant. At the tissue scale, alveolar septa create vast surface area. Smooth muscle regulates airway diameter. At the organ scale, lungs expand and contract. Blood flows through pulmonary circulation. At the system scale, respiratory centers integrate information and coordinate muscles. At the organism scale, ventilation adjusts to maintain homeostasis during rest, exercise, sleep, and stress. Each scale influences the others. Molecular events determine cellular function. Cellular properties determine tissue behavior. Tissue organization creates organ capabilities. Organ performance meets system demands. System regulation maintains organism viability. Understanding respiratory function requires appreciating these multiple levels of organization and their interactions. As you continue breathing, as this exploration continues, the processes we have discussed are occurring within you. Oxygen is diffusing across respiratory membranes, binding to hemoglobin, being carried to tissues. Carbon dioxide is traveling the reverse path. Your brainstem is monitoring chemical signals, adjusting motor neuron firing, changing the depth and rate of your breathing to match your current needs. These processes continue seamlessly, reliably, moment after moment. The respiratory system's beauty lies partly in this reliability, this constant operation that requires no conscious attention, yet responds instantly when demands change. Whether you are reading, walking, running, sleeping, the respiratory system adjusts appropriately. It is a faithful servant that works ceaselessly from birth until death, asking nothing, providing everything necessary for oxidative metabolism and therefore for life itself. We have journeyed through structures and mechanics, through chemistry and control, and now we arrive at a deeper level of understanding, where separate elements unite into integrated function. The respiratory system does not exist in isolation, but rather forms part of a larger whole, interconnected with every other system in the body, responsive to internal and external environments, adapting continuously to maintain the delicate balance that defines health. Consider for a moment the relationship between respiration and circulation, a partnership so essential that neither system can function without the other. The heart pumps blood, but without the lungs to oxygenate that blood and remove carbon dioxide, the circulation would merely move deoxygenated blood in futile circles. The lungs exchange gases, but without the heart to bring blood to the pulmonary capillaries and carry oxygenated blood to the tissues, gas exchange would accomplish nothing. These two systems are functionally inseparable. The pulmonary circulation itself represents this integration beautifully. Blood exits the right ventricle through the pulmonary trunk, which divides into left and right pulmonary arteries. These arteries branch repeatedly, following the branching pattern of the bronchial tree, until they form the dense capillary networks surrounding each alveolus. This parallel branching ensures that air distribution and blood distribution match anatomically, promoting good ventilation-perfusion relationships. The pulmonary circulation differs fundamentally from the systemic circulation in its pressures and resistances. Where systemic arterial pressure averages about 120 over 80 millimeters of mercury, pulmonary arterial pressure averages only about 25 over 8 millimeters of mercury. The pulmonary circulation is a low-pressure, low-resistance system. The pulmonary arteries have thinner walls than systemic arteries of comparable size, less smooth muscle, more distensibility. This low pressure serves important purposes. It reduces the work the right ventricle must perform to pump blood through the lungs. It minimizes the hydrostatic pressure tending to push fluid out of pulmonary capillaries, helping to keep alveoli free of edema. The thin-walled, delicate alveolar capillary membrane could not withstand high pressures without damage. The pulmonary circulation's design reflects the principle that form follows function. The structure is precisely suited to the task. The pulmonary circulation also has unique regulatory properties. We mentioned earlier that pulmonary blood vessels constrict in response to low alveolar oxygen, called hypoxic pulmonary vasoconstriction. This response is opposite to what occurs in systemic vessels, where low oxygen causes vasodilation. The reversed response makes physiological sense. In systemic tissues, low oxygen indicates high metabolic activity requiring more blood flow. In the lungs, low alveolar oxygen indicates poor ventilation and directing blood away from poorly ventilated regions toward better ventilated regions improves overall gas exchange efficiency. This local matching mechanism operates on a regional level throughout the lungs. Well-ventilated areas receive more blood flow. Poorly ventilated areas receive less. If you lie on your side, gravity causes more blood to flow to the dependent lung, but ventilation also becomes greater in that lung due to mechanical factors, maintaining reasonable VQ matching. The system self-adjusts continuously, optimizing gas exchange without requiring any conscious control or central coordination. The volume of blood in the pulmonary circulation at any moment is about 500 milliliters, roughly 10% of total blood volume. This pulmonary blood volume can vary, serving as a reservoir. During exercise, when cardiac output increases, pulmonary blood volume may decrease slightly as blood is mobilized. During certain pathological states, pulmonary blood volume may increase, potentially contributing to pulmonary congestion. The transit time of blood through pulmonary capillaries, about 3/4 of a second at rest, decreases during exercise when cardiac output rises and blood flows faster. Even at the shortest transit times during maximal exercise, gas exchange remains complete in healthy lungs because the system normally achieves equilibration in about 1/4 of a second, leaving a threefold safety margin. This reserve capacity means that even when demands increase dramatically, the respiratory system can still maintain adequate oxygenation. The left atrium receives oxygenated blood from the pulmonary veins, typically 4 veins draining the lungs, 2 from each side. This blood has an oxygen partial pressure of about 100 millimeters of mercury and is nearly fully saturated with oxygen. However, by the time this blood reaches the systemic arteries, oxygen partial pressure has dropped slightly to about 95 millimeters of mercury and saturation to about 97%. This small decrease occurs because not all blood flowing through the left heart came through the pulmonary capillaries and became fully oxygenated. A small amount of venous blood enters the left side directly. Some bronchial venous blood drains into pulmonary veins rather than returning to the right heart. A small amount of coronary venous blood drains directly into the left ventricle through tiny Thebesian veins. This admixture of deoxygenated blood with oxygenated blood creates a small physiological shunt that accounts for the slight reduction in arterial oxygen content. The concept of shunt is important in understanding respiratory physiology and pathophysiology. A shunt occurs when blood bypasses the gas exchange surfaces and enters the arterial system without being oxygenated. The small physiological shunt just described is normal and unavoidable. Pathological shunts, much larger, can occur with certain congenital heart defects or when blood flows through completely unventilated regions of lung, as might occur with pneumonia filling alveoli with fluid or with collapse of a lung region. Shunt blood reduces arterial oxygen content and cannot be corrected by administering supplemental oxygen because the shunted blood never comes into contact with alveolar air regardless of how high alveolar oxygen partial pressure rises. This distinguishes shunt from VQ mismatch, where poorly ventilated but still somewhat ventilated regions can show improved oxygenation with supplemental oxygen. Understanding these distinctions helps clinicians interpret blood gases and choose appropriate treatments. The integration of respiratory and cardiovascular function becomes particularly apparent during exercise. As muscular work increases, metabolic rate rises, oxygen consumption increases, and carbon dioxide production increases proportionally. The body must increase both cardiac output and ventilation to meet these increased demands. The increases are closely matched, maintaining arterial blood gases within normal ranges even during intense exertion. During moderate exercise, cardiac output may increase from a resting 5 liters per minute to 15 or 20 liters per minute. Ventilation increases proportionally from a resting 6 to 8 liters per minute to 40, 60, or even higher. The increase in ventilation occurs through both increased tidal volume and increased breathing rate. The cardiovascular increase occurs through both increased heart rate and increased stroke volume. Remarkably, despite these dramatic increases in blood flow and ventilation, arterial oxygen partial pressure remains near 100 millimeters of mercury, and carbon dioxide partial pressure remains near 40 millimeters of mercury during moderate exercise. The system increases supply to match demand so precisely that the chemical composition of arterial blood barely changes. This represents extraordinary regulatory coordination. The mechanisms coordinating cardiovascular and respiratory responses to exercise are complex and incompletely understood. Chemical feedback from chemoreceptors plays a role, but cannot fully explain the immediate ventilatory response that begins within seconds of starting exercise, before blood gases have had time to change significantly. Mechanoreceptors in muscles and joints send signals that contribute to the exercise hyperpnea. Central command The brain's anticipation of exercise needs may also play a role, increasing ventilation in parallel with the motor commands that activate skeletal muscles. At higher exercise intensities, approaching maximal effort, arterial oxygen may finally begin to decrease slightly, and carbon dioxide may increase. Lactic acid produced by anaerobic metabolism in muscles causes metabolic acidosis, further stimulating ventilation. The respiratory system, even with its substantial reserve capacity, eventually reaches limits beyond which it cannot increase further without excessive work of breathing. Elite endurance athletes may actually experience exercise-induced arterial hypoxemia, where oxygen partial pressure drops noticeably during maximal exertion, likely due to VQ mismatch developing at very high cardiac outputs or to diffusion limitation when capillary transit times become extremely short. For most people, though, the cardiovascular system limits exercise capacity before the respiratory system does. The heart's ability to increase cardiac output typically becomes the bottleneck. Breathing may feel labored during intense exercise, but this reflects increased work rather than inadequate capacity. The sensation of dyspnea during exercise is normal and does not indicate respiratory failure, but rather the body's accurate perception of increased respiratory effort. The respiratory system's role in thermoregulation represents another aspect of integration. Breathing eliminates not only carbon dioxide, but also water vapor and with it, heat. Each breath exhaled carries away both the latent heat of vaporization required to convert liquid water to vapor and the heat used to warm the exhaled air to body temperature. In hot environments, increased breathing contributes to heat dissipation. In cold environments, the respiratory tract must warm and humidify large volumes of very cold, dry air, which can represent a significant thermal challenge. Animals that rely heavily on evaporative cooling through the respiratory tract, such as dogs that pant when hot, demonstrate an extreme version of this thermal integration. Humans rely more on sweating for cooling, but respiratory heat loss remains significant. During exercise, when both metabolic heat production and ventilation increase dramatically, respiratory heat loss can account for a meaningful fraction of total heat elimination. The respiratory system also plays a surprising role in metabolic functions beyond gas exchange. The pulmonary endothelium, the cells lining the pulmonary blood vessels, performs important metabolic conversions. Angiotensin-converting enzyme, located on pulmonary endothelial cells, converts angiotensin I to angiotensin II, a key step in the renin-angiotensin system that regulates blood pressure and fluid balance. The lungs thus participate directly in blood pressure regulation through this enzymatic activity. The lungs also filter blood, removing small clots, aggregates of platelets, and other particulate matter before they can reach the systemic circulation and potentially cause damage in the brain or other organs. The dense pulmonary capillary network acts as a filter, and the lungs have fibrinolytic systems that can dissolve small clots. This protective filtering function operates continuously, silently preventing embolic events that could otherwise threaten health. Immune function integrates closely with respiratory structure and physiology. The respiratory tract is constantly exposed to environmental air containing microorganisms, allergens, and particulates. Multiple defense mechanisms protect against infection and injury. The mucociliary escalator continually sweeps debris upward and out. Mucus contains antimicrobial proteins, immunoglobulins, and other protective factors. The epithelial cells themselves form a barrier and secrete antimicrobial peptides. Deeper in the respiratory tract, in the alveoli, specialized immune cells called alveolar macrophages patrol the air-blood interface. These cells can phagocytose bacteria and other foreign particles, removing them before they can cause infection. The lungs also contain lymphoid tissue and can mount adaptive immune responses, producing antibodies specific to inhaled pathogens. The respiratory immune system must balance protection against threats with tolerance of harmless environmental exposures, avoiding excessive inflammation that could damage delicate gas exchange surfaces. The voice, produced by the larynx, represents yet another integrated function. Speech requires precise coordination of breathing, phonation, and articulation. To speak, we exhale in a controlled manner, passing air through the larynx, where vocal fold vibration creates sound. The pitch of the voice depends on vocal fold tension and vibration rate. The volume depends on the force of exhalation and the amplitude of vibration. The qualities that make each voice unique depend on individual variations in laryngeal anatomy and the resonance characteristics of the pharynx, oral cavity, and nasal cavity. Speaking modifies the normal breathing pattern. We take relatively quick inspirations and then exhale slowly over several seconds while producing phrases or sentences. The respiratory control centers must integrate these voluntary speech demands with the ongoing metabolic demands for gas exchange. Somehow, the system manages both tasks simultaneously, allowing us to speak for extended periods without becoming hypoxic or hypercapnic. The integration is so seamless that we rarely think about breathing while talking unless we attempt to speak very long sentences without pausing. Singing takes this integration even further, requiring sustained controlled exhalation, precise pitch control, and often large lung volumes to support long phrases. Professional singers develop remarkable conscious control over respiratory muscles, learning to engage the diaphragm and abdominal muscles in coordinated patterns that optimize breath support. The respiratory system's dual nature, automatic yet controllable, makes such skilled performances possible. The sense of smell also depends on breathing. Odorant molecules must reach the olfactory epithelium in the upper nasal cavity to be detected. This happens primarily during inhalation, when air is drawn through the nose and eddies up to the olfactory region. Sniffing, a sharp inhalation through the nose, creates turbulent flow that brings more odorants to the olfactory receptors. The respiratory system thus provides not only oxygen, but also the sensory information that allows us to detect and discriminate thousands of different scents. Anatomical integration appears throughout the respiratory system. The pharynx serves both respiratory and digestive systems, a structural efficiency that requires coordinated control to prevent aspiration. The larynx protects the lower airways during swallowing while also enabling phonation. The ribs provide both structural protection for the lungs and serve as the attachment points for respiratory muscles, their mobility enabling breathing movements. The diaphragm separates thoracic and abdominal cavities while serving as the primary muscle of inspiration. The pleural space represents an elegant anatomical solution to a mechanical challenge. It couples lung movement to chest wall movement while allowing the smooth sliding necessary for breathing. The negative intrapleural pressure keeps the lungs inflated while also creating a small amount of tension that helps distribute ventilation. The pleural fluid surface tension holds the visceral and parietal layers together while lubricating their movement. This design achieves multiple functions simultaneously through simple physical principles. Age brings changes to all body systems, including the respiratory system. In infants, the thorax is relatively more compliant than the lungs, opposite to the adult situation. This means that the inward elastic recoil of the lungs is less opposed by outward chest wall recoil, resulting in lower functional residual capacity. Infants therefore have less respiratory reserve and can develop hypoxemia more quickly if ventilation is compromised. The respiratory system matures through childhood. Lung volumes increase with body size. The chest wall becomes stiffer. The number of alveoli increases through early childhood, reaching the adult number by around age 8, though the alveoli continue to grow in size through adolescence. Respiratory control mechanisms mature, with breathing patterns becoming more regular and stable. In healthy adults, pulmonary function reaches its peak in the 20s and then gradually declines. Lung elastic recoil decreases with age, meaning the lungs become more distensible. Chest wall compliance decreases, the ribs becoming stiffer and costal cartilages calcifying. These opposing changes in lung and chest wall mechanics partly offset each other, but residual volume tends to increase with age while vital capacity decreases. The alveolar capillary membrane thickens slightly with age. Alveolar walls lose some elastic fibers. The total alveolar surface area may decrease slightly. These structural changes reduce diffusion capacity somewhat. Pulmonary capillary blood volume decreases. However, in healthy older adults, these changes rarely cause significant symptoms at rest or during moderate activity because the respiratory system's reserve capacity is so substantial that even with age-related decline, adequate function is maintained. Chemoreceptor sensitivity may decrease with age, reducing the ventilatory response to hypoxia and hypercapnia. Older adults may therefore not increase breathing as much in response to low oxygen or high carbon dioxide. Protective reflexes like cough may become less effective. Immune function in the lungs may decline, potentially increasing infection susceptibility. Despite these changes, many older adults maintain excellent respiratory function well into advanced age, particularly if they avoid smoking and maintain physical fitness. Environmental factors profoundly influence respiratory health. Air quality, occupational exposures, smoking, infections, all these leave their marks on the respiratory system. The lungs' constant exposure to the external environment makes them vulnerable to inhaled toxins and pathogens, yet also drives the evolution of robust defense mechanisms. Altitude provides a natural experiment in respiratory adaptation. As elevation increases, atmospheric pressure decreases, and with it, the partial pressure of oxygen in inspired air. At high altitudes, even though the percentage of oxygen in air remains 21%, the total pressure is lower, so oxygen partial pressure decreases. At 3,000 meters elevation, inspired oxygen partial pressure is roughly 30% lower than at sea level. The body responds to high altitude through both immediate and long-term adaptations. Immediately, peripheral chemoreceptors detect low arterial oxygen and stimulate increased ventilation. Hyperventilation raises alveolar oxygen partial pressure somewhat, improving oxygenation. However, hyperventilation also blows off carbon dioxide, potentially causing respiratory alkalosis. Over days to weeks, the kidneys compensate by excreting bicarbonate, allowing pH to return toward normal while maintaining the increased ventilation. Longer-term adaptations to altitude include increased red blood cell production, stimulated by erythropoietin released in response to tissue hypoxia. Higher red blood cell counts increase blood oxygen carrying capacity, partially compensating for lower arterial oxygen partial pressure. Capillary density may increase in tissues, reducing diffusion distances. Cellular adaptations may improve oxygen utilization efficiency. People born and raised at high altitude show these adaptations most fully, with larger lung volumes, greater pulmonary capillary surface area, and other modifications that improve function in low oxygen environments. High-altitude pulmonary edema, a potentially serious condition that can develop in some people ascending to high elevation, demonstrates the respiratory system's vulnerability when stressed beyond its adaptive capacity. The mechanism involves pulmonary vasoconstriction in response to hypoxia. If this vasoconstriction is uneven, some regions developing much higher pressures than others, fluid can be forced from capillaries into alveoli. The fluid-filled alveoli cannot participate in gas exchange, creating a vicious cycle where oxygenation worsens, hypoxic vasoconstriction intensifies, and edema progresses. Descent to lower altitude, allowing reoxygenation and reversal of the hypoxic pulmonary vasoconstriction, is the definitive treatment. Diving, particularly breath-hold diving, presents opposite challenges. As a diver descends, the pressure surrounding the body increases, compressing gas-filled spaces, including the lungs. The total lung capacity decreases proportional to the pressure increase according to Boyle's law. Very deep breath-hold dives require extraordinary skill and physiological adaptation. With the lungs compressed to volumes approaching residual volume, diving while breathing compressed air from scuba gear introduces different issues. The increased partial pressures of gases at depth can have physiological effects. Nitrogen at high partial pressure can cause narcosis, impairment of mental function, sometimes called rapture of the deep. Oxygen at very high partial pressure can cause toxicity, affecting the central nervous system. Ascending too quickly after breathing compressed air at depth can cause dissolved nitrogen to come out of solution in the blood and tissues, forming bubbles that cause decompression sickness. These extreme environments, altitude and depth, reveal the respiratory system's normal operating range and its limits. The system functions optimally near sea level, where atmospheric pressure and oxygen partial pressure match the conditions under which human physiology evolved. Venturing far from these conditions challenges the system's adaptive capacity and can lead to dysfunction if limits are exceeded. Pathological conditions affecting the respiratory system can be broadly categorized by whether they primarily affect ventilation, diffusion, or perfusion, though many conditions affect multiple aspects. Understanding normal integrated function provides the foundation for understanding how disease disrupts this integration and causes symptoms. Obstructive lung diseases, including asthma, chronic bronchitis, and emphysema, share the common feature of increased airway resistance. In asthma, smooth muscle in bronchioles constricts, airway walls become inflamed and swollen, and mucus production increases. All these factors narrow the airways, increasing resistance. The increased resistance makes breathing more difficult, particularly exhalation, which becomes prolonged and effortful. Air may become trapped in alveoli, increasing residual volume and reducing the efficiency of each breath. Emphysema involves destruction of alveolar walls, reducing surface area for gas exchange and destroying the elastic tissue that normally provides lung recoil. The loss of elastic recoil causes airways to collapse more easily during exhalation, trapping air and leading to hyperinflation. Patients develop a characteristic barrel-chested appearance as the thorax becomes chronically overinflated. The work of breathing increases because breathing occurs at larger lung volumes where compliance is reduced. Restrictive lung diseases limit lung expansion, reducing lung volumes and capacities. Pulmonary fibrosis, where lung tissue becomes scarred and stiff, decreases lung compliance. More muscular effort is required to inflate the stiff lungs. Breathing becomes rapid and shallow because deep breaths require excessive work. Diffusion capacity may also be reduced because fibrotic tissue thickens the alveolar capillary membrane. Vascular disorders affecting the pulmonary circulation can impair gas exchange by reducing perfusion. Pulmonary embolism, where blood clots lodge in pulmonary arteries, blocks blood flow to affected lung regions. The blocked areas remain ventilated but unperfused, creating alveolar dead space where ventilation is wasted because no blood is available for gas exchange. Large pulmonary emboli can also cause right heart strain because the right ventricle must pump against increased resistance. Pulmonary edema, accumulation of fluid in the lungs, impairs gas exchange by increasing diffusion distance and potentially flooding alveoli. Cardiogenic pulmonary edema occurs when left heart failure causes blood to back up into the pulmonary veins, raising pulmonary capillary pressure and forcing fluid into the interstitium and alveoli. Non-cardiogenic pulmonary edema can result from increased capillary permeability due to inflammation or injury. Pneumonia, infection of lung tissue, causes alveoli to fill with inflammatory fluid, white blood cells, and debris. The fluid-filled alveoli cannot participate in gas exchange, creating shunt. Pneumonia can also cause pleural inflammation, resulting in pleurisy, painful inflammation of the pleural layers. The pain increases with breathing, leading patients to take shallow breaths to minimize discomfort, which can further impair gas exchange. Understanding respiratory pathophysiology enhances appreciation of normal function. Each disease reveals what happens when one aspect of the integrated system fails. The consequences of failure underscore the importance of each component. The body's attempts to compensate for dysfunction demonstrate the system's adaptability and reserve capacity. The respiratory system's integration extends to its relationship with human experience and consciousness. Breathing is unique among autonomic functions in its accessibility to conscious awareness and voluntary control. We cannot directly control our heart rate, cannot consciously direct peristalsis or hormone secretion, but we can hold our breath, breathe faster, breathe deeper, or breathe in specific patterns at will. This dual nature of breathing, automatic yet controllable, has been recognized across cultures and incorporated into practices like meditation, yoga, and breath work. Deliberate breathing exercises can influence autonomic tone, shifting the balance between sympathetic and parasympathetic activity. Slow, deep breathing tends to increase parasympathetic tone, promoting relaxation and reducing stress responses. Rapid breathing can increase arousal and sympathetic tone. The relationship works in both directions. Emotional states influence breathing, and breathing influences emotional states. Anxiety typically quickens and shallows breathing. Calm contentment allows slow, regular breathing. By consciously slowing breathing, one can trigger the physiological signs of relaxation, which may then feed back to reduce subjective anxiety. This mind-body integration through breathing provides a tangible connection between conscious intention and physiological state. The rhythm of breathing also provides a temporal structure that can serve as a focus for attention. Many meditation practices use breath awareness, attending to the sensations of breathing without trying to control it, as an anchor for the wandering mind. The breath is always present, always accessible, always changing, yet always reliable. It serves as a bridge between the practitioner and the present moment. Even sleep, that altered state of consciousness we enter daily, involves changes in respiratory control. During sleep, particularly during REM sleep, voluntary control of breathing is reduced, and dependence on automatic control increases. Breathing becomes more variable, more influenced by central nervous system activity associated with dreaming. The upper airway muscles may lose some tone, potentially leading to narrowing and increased resistance. In susceptible individuals, this can result in obstructive sleep apnea, where the airway collapses repeatedly during sleep, interrupting breathing and fragmenting sleep quality. The respiratory system in sleep demonstrates the shifts in control that occur across different states of consciousness. Waking allows voluntary override of automatic breathing. Light sleep maintains automatic control with some continuing influence of cortical activity. Deep sleep and REM sleep show different patterns of respiratory control, with chemoreceptor sensitivity changing and breathing patterns reflecting the altered brain states. Throughout all these explorations of integration, with circulation, with metabolism, with immunity, with aging, with environment, with consciousness, we see the respiratory system not as an isolated mechanical apparatus, but as a fully integrated participant in the totality of physiological function. Every breath connects you to the atmosphere. Every gas exchange supports cellular metabolism. Every adjustment of ventilation maintains chemical balance. Every integration enables life. The respiratory system's elegance lies in this integration, this seamless cooperation with other systems, this ability to serve multiple functions simultaneously. It exchanges gases, yes, but also regulates pH, enables speech, provides olfaction, filters blood, contributes to immunity, influences emotional state, and connects autonomic and voluntary control. It is a model of efficiency, of multifunctionality, of elegant biological engineering. As we prepare to move into the final portion of this exploration, take a moment to notice your breath once more. Feel the air moving in and out. Recognize that this simple act of breathing represents the culmination of millions of years of evolutionary refinement, the coordinated action of multiple organ systems, the precise regulation of chemical balance, the interface between your internal world and the external atmosphere. Each breath is a small miracle of integration, a moment of exchange that sustains your existence and connects you to all living things that share this planet's oxygen-rich atmosphere. Part 4. Synthesis. As we move through this final passage of our exploration, the respiratory system reveals itself not merely as a collection of parts or processes, but as a complete living architecture, a system whose whole exceeds the sum of its components. We have examined structures from trachea to alveoli, traced the path of oxygen from atmosphere to mitochondria, understood the controls that adjust each breath to metabolic needs. Now we step back to see the entire landscape, to appreciate how these elements unite into something greater, something that has sustained human life throughout our species' existence. The evolutionary perspective offers profound insight into respiratory design. The transition from water to land, occurring hundreds of millions of years ago, required fundamental changes in gas exchange mechanisms. Aquatic animals extract dissolved oxygen from water using gills. Structures with thin membranes and large surface areas exposed to flowing water. The density and oxygen content of water, however, limit this approach on land. Air contains far more oxygen per unit volume than water, but air's low density means it provides no structural support, and its dryness threatens to desiccate exposed membranes. The lung evolved as an internalized gas exchange surface, protected within the body where humidity can be maintained and delicate membranes shielded from damage. The branching airway system distributes air throughout this internal surface while conditioning it along the way. The pleural arrangement allows the lungs to expand and contract without friction while maintaining their inflation. The integration with a closed circulatory system ensures efficient gas transport to and from exchange surfaces. Different vertebrate groups evolved lungs independently and show variations on this basic theme. Birds, whose high metabolic rates and flight demands require exceptional respiratory efficiency, developed a flow-through lung design where air moves in one direction through rigid tubes rather than in and out of expandable sacs. This allows for continuous gas exchange during both inspiration and expiration, achieving higher efficiency than the mammalian tidal breathing pattern. Air sacs extending from the avian lungs into the bones and throughout the body reduce weight while contributing to this one-way flow system. Mammals, including humans, retained the tidal breathing pattern where air flows into and out of the same passages. While less efficient than the avian system in some respects, mammalian lungs provide adequate performance for terrestrial life and are simpler in design. The enormous alveolar surface area, the thin respiratory membrane, and the close matching of ventilation and perfusion achieve very effective gas exchange despite the bidirectional airflow. The fact that humans can survive and thrive from sea level to altitudes above 5,000 meters in environments ranging from tropical heat to Arctic cold, in dry deserts and humid rainforests, testifies to the respiratory system's adaptability. This adaptability emerges from multiple levels of regulation and compensation. Immediate responses adjust breathing rate and depth. Short-term acclimatization modifies chemoreceptor sensitivity and begins increasing red blood cell production. Long-term adaptation can reshape lung structure itself, particularly during developmental periods. Consider the newborn's first breath, that moment when the respiratory system begins its lifetime of work. In the womb, the lungs were fluid-filled, unexpanded, non-functional for gas exchange. Oxygen came from the placenta, transferred from maternal blood to fetal blood across the placental barrier. The fetal circulation included shunts that bypassed the lungs, directing most cardiac output away from these non-functional organs. At birth, the umbilical cord is cut, eliminating the placental oxygen supply. The newborn must begin breathing within seconds or face hypoxia and death. Simultaneously, the fluid-filled lungs must transition to air-filled organs. The first breath requires tremendous effort, overcoming the surface tension of fluid-filled alveoli and inflating lungs that have never expanded. Once that first breath succeeds, subsequent breaths become easier as surfactant spreads across alveolar surfaces and functional residual capacity is established. The circulatory changes accompanying birth are equally dramatic. The foramen oval, an opening between the atria that allowed blood to bypass the lungs in fetal life, closes as pressure relationships change. The ductus arteriosus, a vessel connecting the pulmonary artery to the aorta and allowing most right ventricular output to bypass the lungs, constricts and eventually closes. Within hours of birth, the circulatory pattern has transformed from the fetal configuration to the adult pattern, where all cardiac output flows through the lungs. This transition from fetal to neonatal physiology represents one of the most remarkable physiological transformations in human development. The respiratory system, which was entirely non-functional for gas exchange, becomes the sole means of oxygenation within moments. The coordination required, mechanical, circulatory, neurological, is extraordinary. The fact that this transition succeeds in the vast majority of births speaks to the robustness of the design. Throughout childhood and into adulthood, the respiratory system continues adapting. Physical training increases respiratory efficiency and endurance. The respiratory muscles strengthen, allowing sustained ventilation at higher levels. Lung volumes may increase somewhat. The body learns to tolerate higher levels of carbon dioxide and hydrogen ions, allowing athletes to maintain intense effort despite the metabolic acidosis of anaerobic exercise. These trainable aspects demonstrate the system's plasticity, its capacity to improve with use. The respiratory system also demonstrates remarkable resilience and reserve. Humans can survive with one lung removed, the remaining lung providing adequate gas exchange even during moderate activity. Portions of lungs can be damaged or destroyed without causing symptoms at rest because the undamaged regions compensate. This redundancy and reserve capacity provides a buffer against injury and disease, allowing continued function even when some components fail. The concept of homeostasis, the maintenance of stable internal conditions despite external variations, finds clear expression in respiratory function. The body maintains arterial oxygen partial pressure near 100 millimeters of mercury and carbon dioxide partial pressure near 40 millimeters of mercury across a wide range of conditions. Metabolic rate varies enormously between rest and maximal exercise. Atmospheric pressure changes with altitude. Air temperature and humidity fluctuate. Yet through all these variations, the regulatory systems adjust breathing to maintain stable blood gases. This regulatory stability emerges from multiple feedback loops operating at different timescales. Breath-to-breath control responds within seconds to chemical stimuli. Acclimatization processes unfold over days to weeks. Developmental and adaptive changes occur over months to years. The multi-layered control provides both immediate responsiveness and long-term adaptation, creating a system that is both stable and flexible. The respiratory system's role in acid-base homeostasis exemplifies the stabilizing function. Metabolic processes constantly generate acids, particularly carbonic acid from carbon dioxide and various organic acids from metabolism. Without regulation, these acids would quickly lower blood pH to levels incompatible with life. The bicarbonate buffer system, with carbon dioxide and bicarbonate in equilibrium with carbonic acid, provides chemical buffering. The respiratory system provides physiological buffering by adjusting ventilation to control carbon dioxide levels, and thus carbonic acid levels. The kidneys provide the third component of pH regulation, excreting hydrogen ions and retaining or excreting bicarbonate as needed to maintain balance. These three mechanisms, chemical buffering, respiratory compensation, and renal compensation, work together. The buffers act immediately but have limited capacity. The respiratory system responds within minutes and can achieve large changes in ventilation, but cannot completely eliminate acid-base disturbances. The kidneys work slowly over hours to days, but can provide complete compensation by adjusting bicarbonate levels. Together, these mechanisms maintain blood pH between 7.35 and 7.45 despite continuous acid production and occasional acid loads. This precise regulation is essential because enzyme function, protein structure, and cellular processes all depend on pH being maintained within this narrow range. The respiratory system's participation in this vital homeostatic process demonstrates its importance beyond simple gas exchange. The integration of respiratory function with other physiological systems creates interesting phenomena at the intersection of different control mechanisms. Periodic breathing, where ventilation waxes and wanes in a cyclical pattern, can occur at high altitude or in certain disease states. This results from interactions between the delays in the respiratory control system and the altered chemoreceptor sensitivities. The system oscillates, overcompensating first in one direction, then the other, creating a rhythmic pattern rather than steady breathing. Cheney-Stokes respiration, a particular form of periodic breathing seen in some patients with heart failure or neurological conditions, shows a crescendo-decrescendo pattern where breathing gradually increases, then decreases, followed by a period of apnea before the cycle repeats. This pattern reflects the prolonged circulation time from lungs to brain in heart failure, creating a delay in the feedback loop that destabilizes control. Understanding such phenomena requires appreciation of the respiratory system as part of a larger control network subject to the dynamics and instabilities that can affect any feedback control system. The respiratory system's mechanical properties create interesting interactions with the cardiovascular system beyond gas exchange. During inspiration, when intrathoracic pressure becomes negative, this negative pressure is transmitted to the great veins and right atrium, enhancing venous return to the heart. This respiratory variation in venous return causes the heart stroke volume to vary with breathing, increasing during inspiration when venous return is greater. This creates respiratory variation in blood pressure, a normal phenomenon called respiratory sinus arrhythmia when it affects heart rate. These mechanical interactions between breathing and circulation can be clinically significant. Positive pressure ventilation, where air is pushed into the lungs rather than drawn in by negative pressure, reverses the normal pressure relationships. Positive intrathoracic pressure during inspiration can impede venous return rather than enhancing it, potentially reducing cardiac output. Understanding these mechanical interactions is essential for managing patients requiring mechanical ventilatory support. The cough reflex, mentioned earlier briefly, deserves fuller consideration as it represents the respiratory system's most powerful protective mechanism. Coughing begins with a deep inspiration, filling the lungs. The glottis then closes while the expiratory muscles contract forcefully, building pressure in the airways. When the glottis suddenly opens, air explodes outward at velocities that can exceed 100 kilometers per hour. This violent expulsion can dislodge and expel foreign material, mucus, or other irritants from the airways. Sneezing follows a similar pattern, but involves the nasal passages rather than the lower airways. The sneeze reflex protects against nasal irritation, explosively expelling air through the nose and mouth. Both cough and sneeze demonstrate how the respiratory system can temporarily abandon its usual pattern of rhythmic gas exchange to perform protective actions when needed. The neural control systems can override the normal breathing pattern to execute these complex motor programs. Hiccups represent an involuntary respiratory phenomenon whose purpose, if any, remains unclear. Hiccups involve sudden involuntary contraction of the diaphragm, followed by abrupt closure of the glottis about 35 milliseconds later, creating the characteristic sound. Hiccups may result from irritation of the phrenic nerve or from central nervous system stimulation of the hiccup reflex arc. While usually benign and self-limiting, persistent hiccups can occasionally indicate neurological or metabolic disturbances. These various respiratory phenomena, normal breathing, coughing, sneezing, hiccups, speech, singing, all employ the same anatomical structures but in different patterns. The versatility of the respiratory system in executing these different motor programs while maintaining its primary function of gas exchange demonstrates sophisticated neural control and mechanical flexibility. The sensation of breathlessness or dyspnea deserves consideration as it profoundly affects quality of life in respiratory disease. Dyspnea is a subjective experience, an uncomfortable awareness of breathing difficulty. It can arise from multiple sources. Increased work of breathing due to airway obstruction or reduced lung compliance creates a sense of effort. Stimulation of chemoreceptors by hypoxia or hypercapnia creates air hunger, a feeling that breathing is insufficient. Stimulation of mechanoreceptors, particularly when there is dissociation between respiratory effort and the resulting ventilation, contributes to dyspnea. The neural basis of dyspnea involves multiple pathways. Signals from chemoreceptors, mechanoreceptors in airways and lungs, and receptors in respiratory muscles all contribute. The integration of these signals in the brain creates the conscious experience of breathlessness. Interestingly, dyspnea can be partially relieved by factors that do not improve gas exchange, such as cool air flowing across the face, suggesting that sensory input
Understanding
modulates
dyspnea
the perception
as a symptom
of breathing
requires
requires recognizing it as a complex perception influenced by physiological signals, psychological factors, and previous experience. Anxiety amplifies dyspnea. Fear of suffocation can create a vicious cycle where dyspnea causes anxiety, which worsens the sensation of breathlessness. Conversely, calm reassurance and breathing techniques that reduce anxiety can improve dyspnea even without changing the underlying pathophysiology. This mind-body interaction around breathing demonstrates once again the respiratory system's unique position, bridging conscious and unconscious processes. The respiratory system's responses to different atmospheric compositions reveal its adaptability and limits. Oxygen toxicity, occurring when breathing very high oxygen concentrations for extended periods, demonstrates that even essential substances can be harmful in excess. High partial pressures of oxygen generate reactive oxygen species that damage cells, particularly affecting the lungs themselves and the central nervous system. This limits the use of high-concentration oxygen therapy and restricts the depths and durations of diving operations using enriched oxygen mixtures. Carbon monoxide poisoning illustrates a different atmospheric hazard. Carbon monoxide binds to hemoglobin with about 200 times greater affinity than oxygen, forming carboxyhemoglobin that cannot carry oxygen. Even small percentages of carbon monoxide in inspired air can quickly bind a significant fraction of hemoglobin, reducing oxygen carrying capacity and potentially causing tissue hypoxia despite normal arterial oxygen partial pressure. The treatment involves administering high-concentration oxygen to accelerate carbon monoxide elimination and providing hyperbaric oxygen in severe cases to further increase oxygen delivery to tissues. Toxic gases and particulates in air pollution challenge the respiratory system's defense mechanisms. Sulfur dioxide, nitrogen oxides, ozone, and particulate matter can irritate airways, trigger inflammation, and over time contribute to chronic respiratory disease. The respiratory system can clear some exposures through mucociliary clearance and immune mechanisms, but overwhelming or persistent exposures can overcome these defenses. The lungs bear the cumulative burden of a lifetime of breathing, and the quality of air breathed significantly affects respiratory health. Smoking, the most significant modifiable risk factor for respiratory disease, demonstrates the consequences of chronic toxic exposure. Cigarette smoke contains thousands of chemicals, many toxic and carcinogenic. It paralyzes cilia, impairing mucociliary clearance. It induces chronic inflammation. It damages alveolar walls, contributing to emphysema. It promotes mucus production and airway narrowing, contributing to chronic bronchitis. It dramatically increases the risk of lung cancer. The progression from initial smoking to established disease often spans decades, with the lungs deteriorating gradually as damage accumulates. Yet the respiratory system also demonstrates capacity for recovery. Smokers who quit show gradual improvement in lung function, reduction in inflammation, and decreased disease progression. The rate of lung function decline slows to approach that of never smokers. Cancer risk gradually decreases, though remaining elevated compared to never smokers even years after cessation. This capacity for recovery, though incomplete, offers hope and provides strong motivation for smoking cessation. Respiratory infections represent another major challenge to lung health. Viruses, bacteria, fungi, and other pathogens can infect the respiratory tract. The common cold, caused by various viruses, typically affects the upper respiratory tract, causing inflammation of the nasal passages and pharynx. Influenza can affect both upper and lower respiratory tracts, causing more severe symptoms and occasionally progressing to pneumonia. Bacterial pneumonias fill alveoli with inflammatory exudate, impairing gas exchange and potentially leading to severe illness. The COVID-19 pandemic demonstrated how a novel respiratory pathogen can challenge individual and public health. The SARS-CoV-2 virus primarily affects the respiratory system, causing disease ranging from mild upper respiratory symptoms to severe pneumonia and acute respiratory distress syndrome. The pandemic highlighted the respiratory system's central importance to health, the vulnerability created by its constant exposure to the environment, and the global interconnectedness that allows respiratory pathogens to spread rapidly. The immune responses to respiratory infections demonstrate the integration between the respiratory and immune systems. The innate immune responses, mucociliary clearance, antimicrobial proteins, macrophages, provide immediate defense. Adaptive immune responses develop over days, producing specific antibodies and T cells that can clear the infection and provide lasting immunity. Vaccination leverages this adaptive immunity, training the immune system to recognize pathogens without causing disease, providing protection against future exposures. Asthma, affecting hundreds of millions of people worldwide, illustrates how respiratory disease can result from dysregulated immune responses. In asthma, airways become hyperresponsive, reacting excessively to triggers like allergens, exercise, cold air, or irritants. The exaggerated response involves bronchial smooth muscle contraction, airway inflammation, and increased mucus production, all combining to narrow airways and cause difficulty breathing, wheezing, and cough. The episodic nature of asthma, with periods of normal function interrupted by acute exacerbations, demonstrates the respiratory system's capacity to shift between different functional states. Chronic obstructive pulmonary disease, or COPD, primarily caused by smoking but also by other chronic exposures, represents progressive respiratory impairment. The disease combines chronic bronchitis with persistent cough and mucus production, and emphysema with destruction of alveolar walls and loss of elastic recoil. As COPD progresses, patients experience increasing breathlessness, first only with exertion, later even at rest. The disease is generally irreversible, though its progression can be slowed. Advanced COPD creates severe disability, with patients feeling breathless from minimal activities. Respiratory failure, the ultimate consequence of severe respiratory disease, occurs when the lungs cannot adequately oxygenate blood or remove carbon dioxide. Type 1 respiratory failure involves hypoxemia without hypercapnia, typically resulting from VQ mismatch or diffusion impairment. Type 2 respiratory failure involves both hypoxemia and hypercapnia, resulting from inadequate ventilation. Acute respiratory failure develops rapidly and represents a medical emergency requiring immediate support, often including mechanical ventilation. Chronic respiratory failure develops gradually in diseases like COPD, with the body partially compensating through mechanisms like increased red blood cell production. Mechanical ventilation, while potentially life-saving, introduces its own complexities and risks. Positive pressure ventilation, as mentioned earlier, alters normal pressure relationships. Ventilator settings must balance adequate gas exchange against the risk of lung injury from high pressures or volumes. Weaning patients from mechanical ventilation requires ensuring the respiratory muscles can resume their work after a period of mechanical support. The process of respiratory failure, support, and recovery illustrates the interplay between pathophysiology and intervention. Lung transplantation represents the ultimate intervention for end-stage respiratory disease, replacing failed lungs with donor organs. The procedure requires precise surgical technique to connect airways, blood vessels, and ensure adequate function. Immune suppression prevents rejection but increases infection risk. Despite these challenges, lung transplantation can restore life to patients with otherwise fatal respiratory failure, demonstrating modern medicine's capacity to replace even this vital system. Throughout these considerations of disease and intervention, the healthy respiratory system's elegance becomes even more apparent. The seamless function we take for granted represents extraordinary complexity working flawlessly. Each breath, accomplished without thought, represents coordinated action of muscles, nerves, airways, alveoli, blood vessels, and control centers. Disease reveals what we lose when components fail. Treatment attempts to restore what health provides automatically. Research continues to deepen understanding of respiratory biology and to develop new treatments. Studies of respiratory stem cells explore the lungs' regenerative capacity and potential therapeutic applications. Investigation of surfactant biology has led to surfactant replacement therapy for premature infants. Understanding of inflammatory pathways has enabled development of targeted anti-inflammatory treatments for asthma and other conditions. Gene therapy trials explore treating genetic disorders affecting the lungs, like cystic fibrosis. Technological advances create new diagnostic and monitoring capabilities. High-resolution computed tomography can image lung structure in exquisite detail, revealing abnormalities invisible to chest X-rays. Pulmonary function testing has become increasingly sophisticated, measuring not just volumes and flow rates, but also detailed mechanical properties and gas exchange capacity. Exhaled breath analysis can detect volatile compounds that might serve as biomarkers for disease. The integration of artificial intelligence with imaging and clinical data promises improved diagnosis and treatment planning. The respiratory system also contributes to our understanding of fundamental biological principles. The study of gas diffusion, ventilation-perfusion relationships, and respiratory control has illuminated principles of mass transport, feedback regulation, and physiological integration applicable throughout biology. The respiratory system serves as a model for studying how complex biological systems achieve stable function, how they respond to perturbation, and how structure determines function. As we near the end of this exploration, we return to where we began, to the simple act of breathing, to the rise and fall of the chest, to the quiet passage of air. But now this simple act reveals its depths. We understand the anatomy that channels air, the chemistry that exchanges gases, the physics that drives flow, the regulation that adjusts ventilation, the integration that connects breathing to all of physiology, the evolution that shaped this system, the development that brings it into function, the diseases that can impair it, the treatments that can restore it. The respiratory system is ancient, its basic design established hundreds of millions of years ago. Yet it is also uniquely yours, shaped by your genetic inheritance, your developmental history, your environment, your habits. Your lungs have taken every breath you have ever breathed, have oxygenated your blood during your first cry, and will continue until your last breath. They have supported you through illness and health, through rest and exertion, through all the variations of your life. There is something profound in recognizing that this process continues regardless of our awareness. Right now, as you read these words, as your attention is focused on ideas and concepts, your brainstem is monitoring carbon dioxide levels, adjusting the activity of respiratory motor neurons, contracting and relaxing your diaphragm and intercostal muscles, moving air in and out, sustaining your existence. This automatic operation, this reliable function, this constant background process enables everything else: thought, emotion, movement, sensation, life itself. The respiratory system connects you to the world in a tangible way. The air you breathe is shared with all other breathing creatures. The oxygen molecules entering your lungs might have been released by a tree's photosynthesis, might have cycled through countless organisms over millions of years. The carbon dioxide you exhale will be taken up by plants, incorporated into organic molecules, returned to the atmosphere in an endless cycle. You are not separate from the environment, but constantly exchanging matter and energy with it, participating in planetary scale cycles of carbon and oxygen. This perspective, seeing yourself as part of larger systems, connected through breath to all of life and to the atmosphere itself, can shift understanding of our place in nature. We are not isolated beings, but thoroughly integrated into Earth's biosphere. The respiratory system makes this connection tangible, immediate, continuous. Every breath is an exchange with the world. Indigenous wisdom traditions and contemplative practices across cultures have recognized breathing's significance beyond mere physiology. Breath has been seen as life force, as spirit, as a bridge between body and consciousness. The word for breath and spirit are the same or related in many languages. Pneuma in Greek, prana in Sanskrit, ruach in Hebrew, qi in Chinese. These traditions recognize something essential about breathing: its connection to vitality, to presence, to life itself. Modern science and ancient wisdom converge on the breath's importance, though describing it in different languages. Science speaks of oxygen and carbon dioxide, of partial pressures and diffusion gradients. Wisdom traditions speak of life force and energy. Both recognize breathing as fundamental to existence, and both have developed practices working with breath. Science to understand and treat disease. Contemplative traditions to influence consciousness and well-being. The integration of these perspectives offers richer understanding than either alone. The scientific knowledge we have explored provides detailed mechanistic understanding of respiratory structure and function. Contemplative awareness of breathing provides experiential knowledge of the breath's role in consciousness and well-being. Together, they paint a complete picture of the respiratory system's physical, physiological, and existential significance. As this exploration draws to a close, as we complete this journey through the respiratory system's territories, notice once more your breathing. Notice it not with the intention to change it, but simply to observe it as it is. The pace, the depth, the rhythm, the sensations. Notice how it continues whether observed or not, how it adjusts automatically to your needs, how it connects you to this moment and to the world around you. The respiratory system we have explored is not an abstraction, but a living reality within you right now. The structures we discussed are carrying out their functions at this very moment. The alveoli are exchanging gases. The chemoreceptors are monitoring chemical signals. The respiratory centers are generating rhythmic activity. The integration is happening seamlessly, automatically, reliably. In understanding the respiratory system deeply, we gain not just knowledge but appreciation. Appreciation for the elegance of biological design, for the wisdom of evolutionary adaptation, for the robustness of physiological regulation, for the integration that creates coherent function from myriad components. We recognize the respiratory system not as mere machinery, but as a beautiful expression of life's capacity to organize matter and energy into forms that sustain themselves, that maintain order in the face of entropy, that persist through time. The respiratory system will continue its work long after this exploration ends. It will continue through your sleep tonight, through tomorrow and all the days ahead, adapting to whatever demands arise, maintaining the chemical stability that enables life, connecting you breath by breath to the atmosphere. It asks nothing and gives everything. This faithful system that began with your first breath and will end only with your last. Rest in the knowledge that you are held, sustained, supported by systems of extraordinary sophistication and reliability. Your breathing continues, your blood remains oxygenated, your cells receive what they require. The respiratory system, in concert with all the body's other systems, maintains your existence with precision and grace. There is nothing you must do, nothing you must control. The wisdom built into billions of years of evolution, refined through millions of years of mammalian development, expressed in your particular body, does its work quietly and well. The air moves in and out. The exchange continues. Life persists. And in this simple rhythm, repeated moment by moment, breath by breath, we find one of biology's most essential and elegant achievements. The respiratory system, bridge between organism and atmosphere, foundation of oxidative metabolism, enabler of consciousness, sustainer of life.